And we want to think about regulation of enzyme activity. Now, when we say regulation, what do we mean? Regulation means how do we turn enzymes on or off? How do we determine if to speed up their action, decrease the speed of action, etc. So an easy example to think about why this is important is if you think about the gut enzymes, for example, the pancreatic enzymes that are released into the GI tract, those enzymes are made to degrade tissue, right? They're made to break things down, especially the food that we eat, to liberate the biomolecules we talked about earlier in this packet. And so when we think about that, enzymes could potentially dissolve or degrade the entire body, right? So what prevents enzymes from actually eating human tissue? What prevents enzymes from not just breaking us down entirely, right? And that is what we mean by enzymatic activity and regulating of that activity. Now, there are four mechanisms to regulate enzymatic activity: allosteric, covalent. We're going to talk about feedback inhibition, and the opposite of that being feed-forward activation. Let's start with allosteric regulation. So what does this mean? Allosteric means that we're going to bind some type of modulator. So some type of external compound or element will bind to our enzyme at its regulatory site. So here we're thinking about two different sites for binding on the enzyme, and the modulator binds to the regulatory site, and that has an effect on the configuration of the active site. So that modulator can sort of impact how the enzyme behaves, what it binds with, what type of activity it's going to engage with, and whether it's turned on or turned off. Okay. Likewise, if we remove the modulator, right, we have the regulatory site blank or empty, that may altogether inactivate our enzyme. And so this is how we can turn it on, allosteric regulation. The second mechanism is covalent regulation. So very similar to what we saw with the allosteric regulation. The difference here is that we have another two enzymes that are involved in our enzyme in question. So this is our enzyme in question. But there is another enzyme that can add some chemical group to the enzyme that we are regulating or modulating. And so the chemical group, sometimes a phosphate, but it can be other types of compounds here. The chemical group binds to the enzyme in question with the help or catalyzed by enzyme A. And so this is the state of the enzyme on this side of our image here with this chemical group covalently bound. So now we have altered the enzyme activity with the help of a secondary enzyme, enzyme A, by adding some chemical group to the enzyme in question. Okay. The opposite of that is catalyzing or breaking of that covalent bond, and that is going to return the enzyme in question back to its original state without the chemical group, and that is done by enzyme B. So two other enzymes are involved to help change the state of the enzyme that we're regulating to either turn it on or turn it off, speed it up, slow it down, regulate its activity in some way. A good example of this that we'll talk about is with protein kinase. Protein kinase, when we hear the word kinase, this is kind of a rule of thumb going forward, it is going to add a phosphate, okay? And when it adds a phosphate, it's going to change the catalytic activity of the enzyme that we've added the phosphate to. Alternatively, phosphatase, when we hear that word phosphatase, it's going to cleave or cut the phosphate off. We'll talk about some of these in our next packet in cellular respiration, some of these enzymes. And so phosphatase takes the phosphate group off of the enzyme that's being regulated and again shifts its activity by regulating in that way. Okay, so that is kinase versus phosphatase as examples of enzyme A and enzyme B. Let's talk about, so here's that example, actually, I kind of jumped ahead of myself. So here's that example. Here's our enzyme that's being regulated. Here's the phosphate that we're going to add. Here is protein kinase, which is the enzyme that's going to put that phosphate on our regulated enzyme. And then we have the enzyme phosphate bound. Alternatively, phosphatase will cleave that phosphate off of the enzyme phosphate complex and bring us back to separate enzyme and phosphate. So in this two examples here, the function of the enzyme is going to be different depending on whether it has that phosphate bound or not. All right, the third mechanism is feedback inhibition. And feedback inhibition is a way to modulate enzymes that are upstream, that are downstream, so behind or backward in terms of direction. That's why it's feedback. And the way this happens in our example here is we have enzyme 1. Enzyme 1 catalyzes the reaction of substrate A converted into product B. Product B becomes a substrate for enzyme 2 and catalyzes that to product C. So the rate of enzyme 1 and enzyme 3 are constant, right? We're not going to modulate or regulate those. The rate of enzyme 2 is going to be dependent on the concentration of C. So we can consider this step, B to C, as the rate limiting step. So how much of product C we make will determine the activity of enzyme 2. Let's say we have a lot of C that has been generated and it's sort of building up. That is going to in turn shut off the activity of enzyme 2 and slow the entire reaction down at that step. So this is why we consider this the rate limiting step. All right. Another example of feedback inhibition, in this one, the rate of enzyme 2 and 3 are constant, so these are not being regulated. Instead, the amount of product D is going to determine the activity of enzyme 1. So we can consider A to B as the rate limiting step in this equation because D will determine how fast A to B will proceed or how slow A to B will proceed, right? This is the rate limiting step. Lastly, we have feed forward activation. This is in the opposite direction. This is very rare. There are very few examples of this in our body or in just in general in nature. And so when we think about this, we have regulation that's happening upstream instead of downstream, right? In the backward direction. So this is happening in the forward direction, and it's actually activating. So when we think about product B, right? A is converted to B by enzyme 1. The amount of B is going to determine the activity of enzyme 3. So if we have a lot of product B being produced or created, that will actually increase the enzymatic activity of enzyme 3 and sort of help us to shuttle this excess B that's being created onward or forward in the reaction and help to increase the activity of E3, right? Enzyme 3. Okay. So those are some of the ways that we can regulate enzyme activity. Again, increasing the rate, decreasing the rate, or as we saw with allosteric and covalent regulation, we can turn enzymes on and off depending on what is bound to those enzymes.